Thank you all for joining us today. Welcome to the Society for Marine Mammalogy's monthly Editor Select webinar series, where we have this goal of highlighting the latest exciting research that's in the Marine Mammal Science journal. My name is Teresa. I'm going to be hosting today's webinar. I am not Daryl, who is the editor of Marine Mammal Science, but I'm one of the student members at large for the Society for Marine Mammalogy, who helps to organize these talks. I'm also a PhD student at the University of California, Santa Cruz. So this month, we're thrilled to have Maria Glarou here with us. She's going to be telling us about her research using drone images to study sperm whales. Maria is originally from Greece, and she's currently a PhD fellow at the University of Iceland's Research Center, where she looks into the allometry of physiological and behavioral thermoregulatory adaptations of different sized cetaceans. And we'll ask her some more about that at the end, maybe if we have time. Her main research interests revolve around bioenergetics and ecology and ecophysiology, especially in cetaceans, as well as the impacts of human disturbance on those cetaceans. So just as a reminder for all of you listening, there will be time for questions at the end of this presentation. And the Zoom webinar has a Q&A function that we ask you to use at the bottom of your screen to post any of those questions, which we'll then be able to ask to Maria. And Maria's paper is also available via open access on the Marine Mammal Science website if you want to check out more and see some more of her cool figures and methods. And so with that, I will turn it over to Maria.
Well, thank you, Teresa, for the introduction. Hello and welcome, everyone. I will just close my camera now. So welcome. Welcome to my talk. I will talk to you today about a project that I carried out as part of my master's internship just before I started my PhD. And the title of my talk today is How to Weigh a Sperm Whale Using Drone Images. So I will start by giving a bit of background information. So body mass is a key life history characteristic of animals, and it's shaping most of aspects of their ecology and behavior. And for example, it can influence their basal metabolic rate, their caloric needs, feeding habits, growth, home rates, and lastly, their thermoregulation capabilities. And whales are the largest animals on the planet. And due to their size, they play very important ecosystem roles. However, the role of body mass is often understudied, especially in large whales, because obtaining mass measurements from free-ranging individuals is inherently very, very difficult. And sperm whales are the largest living odontocete and the largest toothed apex predators on the planet. And they exhibit sexual dimorphism, and with females typically reaching about 11 meters in length and up to 15 tons in mass, and males reaching around 16 meters in length and about 45 tons in mass. They are deep diving species, and using a powerful biosonar, they are able to echolocate and catch their prey in the dark deep sea environment, thus playing a very vital role in the deep sea energy turnover. And the large body size of sperm whales enhances their diving capacity and allows for increased energy storage due to the low mass-specific metabolic rates. Despite being a very important characteristic of sperm whales, body mass is seldom included in studies of their ecophysiology and bioenergetics due to the difficulties that I mentioned just before of obtaining direct measurements from live animals. And until now, measuring the body mass has been possible only through physical contact with the animal, basically, through strandics, bycatch, or even whaling. And evidently obtaining measurements from dead specimens, either stranded or caught, comes with certain constraints or ethical concerns, of course. Another way, alternatively, we could get the weight predictions from existing weight length curves, but this, of course, doesn't account for any fluctuation that the animal would have in body volume or condition. So drones, or unoccupied aerial vehicles, offer a new and non-invasive way to estimate the body mass of free-living animals, while also accounting for variation in the body condition. And Christiansen et al. 2019 has developed and applied this method on southern right whales. So yeah, the aim of this present study was to apply the same method on sperm whales to estimate the body mass of free living animals using drone images. And obtaining information on body mass has, of course, important implication for future studies of physiology and bioenergetics. So moving on to the methodology. We used unoccupied area vehicles, as I said, also known as drones, to apply aerial photogrammetry methods. We collected images of sperm whales between 2017 and 2020 in 3 locations around the globe, in Dominica, in the Eastern Caribbean Sea, and then in the Balearic Islands in Spain and the Hellenic Trench in Greece, both of them located in the Mediterranean Sea. For this fieldwork, we used 2 different drone models. One was the DJI Inspire 1 Pro, equipped with the Zemius X5 camera with a 25mm lens, and also a laser rangefinder attached to it. And we also used a DJI Phantom 4 Pro in one occasion with a rangefinder, in another one without a rangefinder. So yeah, using the drone, we are able to collect images of sperm whales from a 90-degree angle, like the one that you see on the right in this figure. These images allow us to see the body contour of the whale and extract pixel measurements of certain morphometric parameters of the whale, for example, its body length. And then knowing the altitude of the drone and some camera parameters and lens parameters, we are able to convert those pixels to meters. And in that way, we can measure certain things on this whale non-invasively. As I mentioned just before, in one occasion, we didn't have the rangefinder on the drone to know the precise altitude. So in that case, it was in the Balearic Islands. And in that case, we used an object of known length to scale the pixels to meters. And this was the boat. Basically, we knew a certain part of the boat, we knew the length, and we had an image of the boat and the whale next to each other, basically, and we were able to scale the measurements that way. So, apart from body length, we also extracted measurements of body widths at every 5% increments across the body, as you can see in the top image. And when the whales were turning and showing their lateral side in the bottom image, we were able to also extract height measurements at the same 5% increments. So from the width and the height measurements, we were able to calculate the height to width ratio at each 5% body segment. And based on those ratios, we were able to basically create a body shape model. And each 5% segment was modeled as a series of infinite ellipses. And then the volume of each segment of this whale was calculated. And then adding up all those segments together, we were able to calculate the total body volume of each whale. So moving on to the next step, to convert the volume to mass, we wanted to compare 4 different approaches. So we created 4 different models. So yeah, I've listed here the models. I will talk in more detail about them in the next few slides. Overall, the mass can be calculated by multiplying basically the volume of the animal with a body density estimate. And then that's the theory behind it. And then with each model, each of the 4 models, we came up with one density estimate for each approach. And then we used to get the body mass estimates at the end. And a correction factor was also applied to all 4 approaches to include also the weight of the gas components of the sperm whale body, so the air that was included in the body. So yeah, for model 1, we assumed a uniform body composition and calculated the mean body density using density estimates from whaling records. So basically what we did is we looked through the literature extensively, and we were able to find 3 animals that were caught in South Africa in 1973. And on those animals, the length, the mass, and the girth at the anterior end of the pectoral fin was measured. So we had three measurements from those animals for each of the three animals. So using then the height to width ratio of the free-ranging whales, the ones we had with the drone, we were able to calculate the circumference of the live animals at the same point where the girth measurements of the caught specimens was taken. And then we modeled the body volume as a function of the total body length and the girth. And using this model then on the dead animals, we were able to predict their volume. So then we had volume and mass estimates for the dead animals, for these three animals. And then the body density of each dead animal was then calculated by dividing the mass with the volume. And then we got a body density for each of the three of them, and then we averaged those three together, and then we used this number to convert the volume to mass for this model, for this approach, for all our drone animals. For the second approach, we used an existing tissue density estimates that was published in a paper in 2004 by Miller et al. And this density estimate was estimated from digital tags that were put on the sperm whales. So for the third approach, model 3, we assumed now a non-homogeneous body composition. So basically the body consists of several tissue types like blubber, muscle, bones, and viscera. And then to account for the different weights of each and different densities for each tissue type, we basically obtained the average body density by using the relative proportion of each tissue type combined with tissue-specific weights and published tissue densities. And we came up with a number for that approach. And then in the last model, model 4, we went back to the whaling records and examined, we want to look at the effect of body size on body density, because volume might have an effect on body density. I will explain a little bit more later. And to do that, basically, we modeled the estimated total body density of the three dead animals as a function of body volume. And then we were able to obtain a volume-specific body density for each free-ranging animal. So it was not one constant body density, but a volume-specific one. It would change for each animal based on their volume. And all of the resulting linear models that we created were compared to 4 previously published mass at length relationship, just to see how they compare with the literature. So moving on to the results. In total, we collected data on 43 days between 2017 and 2020 in all locations. And since our methods were not really dependent on location, the data set from all the places was pulled together. And for any further analysis, we just considered the total data set and moved on with all the animals that we sampled. So after quality filtering, we ended up having 102 aerial images of sperm whales, and of which 23 were classified as calves, 11 were classified as juveniles, 55 as adults, and 13 as mothers. And we also had 7 individuals with matching dorsal and lateral images. So if you remember the first figure that the whale was turning also on the side, we had 7 of them that showed us both the dorsal and the lateral side. And from those animals, we were able to extract the height and width measurements and calculate height to width ratio across the body, and then create a body shape model. So this is the height to width ratio. As I mentioned just before, this is the average height to width ratios across at each 5% body segment. And we can see here that the cross-sectional body shape of sperm whales varies noticeably along the body axis, and the the height to width ratio was always larger than 1. So this suggests a vertically elongated body shape throughout all segments. And this makes sense because sperm whales look a bit elongated vertically anyway. So it fits with the images that we all have about sperm whales. Yeah, and this was especially pronounced towards the end of the body. So beyond 60% body length from the rostrum and backwards, then it was more pronounced, this vertically elongated body shape. And yeah, in contrast, the shape in the middle part of the body, so between around 25% of the body length and 45% of the body length, approached almost a height to width ratio of 1:1, which indicates kind of an assumed circular body shape. And yeah, this 1:1 ratio is represented by the red line in the figure. Yeah, and you can see that it was a bit closer to that ratio in the middle part of the body of the sperm whales. Yeah, so these height to width ratios were based on 7 animals. As I said, 2 of them were calves and 5 of them were adults. And we checked if there was any significant differences between the reproductive classes or even between the locations, and we concluded that there is no significant difference. So we used the same height to width ratios to all the animals and applied the same average height to width ratios to all the animals to calculate their volume. Yeah, and now moving on to the volume, the estimates of sperm whale volume ranged from 0.6 to about 24.3 cubic meters. And yeah, as you can see in the figures, these are it shows the relationship between body volume and body length. The first one shows the log-log relationship. So, and the black line is the fitted regression line. And then the second figure, the one below, shows the back transformed regression line. So yeah, in both those figures, you can see that the body volume seems to fluctuate more in larger whales, like more than 8 meters long. And this, of course, indicates equivalent fluctuations in the relative body condition for those animals. Yeah, and here we see the results for all the 4 approaches when we actually calculate the density, the body density, and the body mass. For model 1, the body density was about 879 kilogram per cubic meters, and the body mass was ranging from 516 to 21,390 kilograms. The second model had a bit higher density, 1,003 more or less kilogram per cubic meters, and the body mass prediction were also higher, 589 from the smallest animal to 24,430 kilograms for the largest one. Model 3 was also similar with model 1. We found close numbers between the 2 models, 887 kilograms per cubic meter for the density, and for the body mass, 521 kilograms to 21,600 kilograms for model 3. And model 4, if you remember, I mentioned that for this one, we didn't have a constant density. We had a varying density based on the volume of the animals, and this ranged from 666 to 978 kilograms per cubic meter. And the body mass predictions were also different than the other models, starting from 575 to 16,240. Yeah, and here I will take each model one by one to see how it compares with the 4 existing mass at length curves from the literature. So basically, we compared all our models with the same 4 curves, and some of them, like the gray one and the green one, were based on whales weighted in parts when they were killed from whaling records. So there was, when the whales are weighted in parts, there is no correction for the fluid that was lost while they are like processing the whales after they kill them. So both these curves are unadjusted for fluid loss. But we also had two more, the yellow one and the blue one, that were adjusted for fluid loss. One of them was adjusted for 10% fluid loss. So all the weight measurements were increased by 10%. And then the other one by 25% fluid loss. So there are some differences in the literature of how everyone is approaching the fluid loss matter. And yeah, you can see that the first model that we had, the first approach, falls closely with the yellow and the green curve. And one is unadjusted for fluid loss, and one is with a 10% adjustment for fluid loss. The second model here now falls closer to the blue line, the blue curve, and this was adjusted for 25% fluid loss. And just to remind you, the model 2 was with body density derived from digital tags. Model 3 now here, we calculated the body density based on the relative proportions of the different tissue types. And this one falls again closely with the 10% adjusted for 10% fluid loss curve and the unadjusted for fluid loss curve. So the yellow and the green one, very similar to model 1. And then model 4 is the one with the lowest weight predictions, and it's the one with the varying density, the volume dependent density. And this one falls very close to the two unadjusted for fluid loss curves. So just to reiterate, now moving on to the discussion, what we did here was use drones to collect images of sperm whales, from which we extracted morphometric measurements and created a body shape model. And using this model, we were able to calculate the volume of the animals using a series of ellipses. And then using 4 different approaches, we were able to estimate total body densities for each approach and convert ultimately the volume calculations to mass estimates. And yeah, our elliptical model provides a closer approximation of the body shape of whales, especially compared to other methods that were used in the other models that were used in the past, like the truncated cones method or the cross-circular body model. Yeah, and we also, as I mentioned, we used 7 animals to build this model on, with the height-width ratio of those animals, but the mean error of using just 7 animals and then predicting the heights of the other animals that only showed us the dorsal side. So we only had images like the one you see here, so not on the lateral side. We used the ratios from the other animals, the 7 animals, to predict the heights of those animals that we didn't get to see the lateral side. And this has a very small error as a method, only 5.4%. Christiansen et al. 2019 already compared it and calculated this error. So this gives us a bit of confidence in the accuracy of our method. Yeah, and overall, all body mass models had strong predictive power. The R-squared was more than 0.97, and all the estimates were in line with published weight measurements and existing weight length relationships from the literature. So yeah, so models 1, 2, and 3. For those ones, we assumed a constant tissue density through the body. So we did not account for the effect of body girth and volume on the body density. So yeah, but in animals with increased volume relative to their length, we can assume that this increase in volume might be because of the extra blubber. So then the percentage of the weight of the other tissues is relatively lower, but still the tissue-specific mass should be rather stable. And because the density of the blubber is lower than the other tissues, we are assuming that the mean body density should also decrease with an increasing volume and vice versa. So the only model that takes this into account is the model 4, the last one, that for which we accounted for the volume dependent changes in the body density. But unfortunately, we had a very small sample size. We based this only on 3 animals, the ones that were caught in South Africa. And yeah, so we couldn't find a significant relationship between density and volume, but still as an idea and as something to consider is very important. That's why we included it also in the paper. And I'm mentioning it now. Yeah, because it might be very valuable for future research or for other species that there might be more data available. So you can find a relationship and hopefully get more accurate predictions of mass. Yeah, so overall, all models resulted in a positively buoyant average total body density. So from 834 to 1003 kilogram per cubic meter. And this is also supported by observation of in the literature of sperm whales that typically float when they are killed. So this is also indicating, of course, positive buoyancy because they are floating. And there are very few rare cases of negatively buoyant sperm whales that have been reported. So those mostly refer to younger animals with less blubber tissue, or to incidents when the air might be lost from the body due to a wound. So then, but the fact that some of the sperm whales have been reported to sink, so they became negatively buoyant, basically, when the air was removed from their body, then this suggests that the higher total body density value of Miller et al. 2004 that was used in model 2 of this study is likely the most accurate compared to the other 3 models, as this is the only approach for which the tissue-only density, so if we don't include the gas, it would be negatively buoyant when we calculated the tissue-only density. So this is probably the closest to reality. Yeah, another consideration is, as I mentioned previously, is the fluid loss that happens during flensing. Flensing is the process of removing the blubber from the rest of the body during post-processing of the animal when they catch it in historical times. So then this fluid loss is estimated to be between 13% and 15%. And overall, the total blood volume of sperm whales is about 20% of its total mass. So yeah, all our models, the ones we built, were all accounting for the weight of the body fluids. So we would expect that none of them would maybe match with or be similar to any of the uncorrected weight at length relationships that we compared it to. But model 3 and 4 were similar to the 2 uncorrected curves. And this might indicate a slight underestimation of the weight prediction from these 2 models. Then on the other hand, model 1 was similar to a curve with 10% fluid loss correction, which is still a bit of a conservative estimate, considering that the fluid loss during flensing might be 13 to 15%. Yeah, so this could also indicate some slightly underestimated weight estimates. Yeah, and then model 2 was very similar to the curve that accounts for 25% fluid loss. So then there might be a slight overestimation of the weight from this model. So yeah, the slightly skewed results of all these models can be attributed to these uncertainties that surround the existing body weight data, such as the fluid loss and the lack of empirical tissue-specific densities, especially for sperm whales specifically, or also on the small sample size of the whaling data that we used for models 1 and 4. So yeah, these inaccuracies in body mass predictions will not affect any relative metrics that we would like to explore. So if someone wants to study the body condition, like this won't be affected, but of course it may bias any mass-specific metrics. So if you want to look at mass-specific metabolic rates, then it will be biased. So therefore, there is a need to have a more empirical measurements of densities of different tissue types and specific to the species in question. And also future studies will need to validate the existing density estimates with methods combining UAV photogrammetry and tissue density estimates from animal-borne tags by applying the both methods on the same individuals. And to sum up, our method is non-invasive. We are able to get all these measurements just by flying a drone over an animal. We don't need to go near it. It's easy to replicate. You need this drone setup that I described before, and yeah, you could replicate the method, and it can be cost efficient, considering other type of research, and especially if you can do it from the land. In some places, you're able to fly the drone. If the whales are close to the land, you can fly it from the land. So it can really bring down the cost a lot. And yeah, it can also be applied to a variety of marine mammals and other taxa even. So it's really widely applicable. Yeah, and of course, for the future, it is important to consider some refinements for this method, such as validating the tissue density estimates and applying an accurate fluid loss correction, especially if working with whaling records. And yeah, while we can recognize these uncertainties around the body density estimates, obtaining non-invasive predictions of body mass of free-ranging animals remains highly valuable for several ecological and physiological questions, and especially given their central role in marine ecosystems. So, for example, our methods can be used to study somatic growth and metabolic rates, intraseasonal or interannual changes in body condition, patterns of fattening and growth, estimating mass-specific cost of reproduction, locomotion, and heat loss. All of these questions, ecological questions, ecological matters, would have big implications for management and conservation, of course, in the future for the species. And yeah, if you want to read more about this project, the article is freely available to download from Marine Mammal Science. And at this point, I'd really like to thank my co-authors and all the funders for making this work possible, as well as the Society for Marine Mammalogy for inviting me to give this talk today. And yeah, of course, thank you all of you for your attention and your time. And I would be happy to take some questions now if there are any. Yeah.
That was great, Maria. Thank you very much.
Thank you.
Always fun to see pictures from drones and then get to learn more about how they're being used.
Indeed.
So yes, just a reminder to our listeners, please post any questions that you have using that Q&A function that's at the bottom of your screen. You see a little Q&A boxes, chat window kind of thing. We would love to know what you might be interested in. I have a couple that I'll ask in the sort of intermediate period while people take some time to get their questions. One's just a little, what I imagine is pretty little, and you talked about a correction for the gas in the animals' bodies. Where does that measurement come from? Is that from using stranded animals or, you know, whaling records? I was just curious.
So, well, yeah, I didn't get into much detail here in this talk. It's more explained on the paper, but basically what we thought that the dead animals, the ones, because we based the two of our models on dead specimens, and those ones, due to the compression of the like gravity, they would have released some of the air. So by reading up on the literature, I was able to maybe estimate that maybe based on the tidal volume, it will be around 90% of the air that they would have, that it would be released due to the compression and the weight. So then we would assume that those ones, the weight that we had from the dead animals would be the tissue only plus 10% of the gas. And then I was able to add like the rest of the 90% because we're working with live animals and we want to apply the same thing on the live ones, that they would have the air on their lungs. So yeah, this was kind kind of the idea behind it to correct for this. And it was just by reading up on tidal volume. There is really not a lot of information. It's really difficult to estimate, but I was thinking about 90% would be correct that it would be missing based on, yeah. It's also, anyway, all of it kind of a estimation, so. Yeah.
Yeah. Well, but it just goes to show, right, why it's so valuable to have these multiple different methods that you can compare, because you have, like you said, the fluid loss or gas losses that happen in certain situations. So related to that, we have a question in the Q&A, which is wanting to know more about how those density estimates are derived from tagging. So the model that you had that talked about using tags.
Yeah, well, I think, yeah, we didn't do that. We just took it based on the paper from Miller et al. 2004. And I think they're able to estimate it based on the drag coefficients and like the acceleration, and they had some length estimation and weight estimation of this animal, how it would be from another method. In 2004, there were no drones, so they were like trying to estimate the length from I think the distance between the dorsal and the head. So they extrapolate the length. Anyway, they had this information, and then by using like, it's not really my specialty, but I think from my understanding, it's because of the drag and how it glides through the water, the animal, and that they are able to, and the speed and everything, and then they're able to come up with a density estimate. It is all very well explained on that paper. Yeah, it's interesting actually.
Perfect. Yeah, that's wonderful. Thank you. Let's see, I'll give folks a chance to ask other questions. You mentioned some sort of major changes in body condition, and I recognize that this was not the focus at all of the study, but I was wondering if you noticed even, kind of anecdotally, any patterns or things that stood out. I think you mentioned that there was some, if I'm remembering correctly, some kind of changes in the body condition that you were able to see in sort of the variation in the model results.
Yeah, what we saw was that the volume was fluctuating a little bit more for the larger animals, which kind of makes sense. They might be in different stages of like, I guess, yeah, in their life at this moment. Also keep in mind that sperm whales are income breeders, so not capital breeders, so they don't have these really clear seasonal changes in condition like, for example, humpback whales would have, or other baleen whales that are capital breeders. So yeah, there are some fluctuations. I didn't really look into that. There are some graphs on the paper, but yeah, it's basically what we saw is that it changes mostly on the larger animals. It's just a lot of fluctuations, not necessarily like it varies between the population, I guess, that we studied. But we didn't do any seasonal monitoring or anything like that. It was, yeah, mostly just examining those ones. How do they look compared to the rest? Yeah, and there was a bit of fluctuation on the larger animals. Yeah,
got it. Well, just goes to show that if I'm understanding correctly, you would be able to capture that kind of fluctuation using this method, like you were talking about, right? So if there's a researcher that wanted to look into that kind of question in more depth, that's absolutely something you could do with this method.
Definitely. Yeah, definitely. Yeah, and this is, you don't even have to go all the way to mass. You can just, the first step, just relative even. Body condition is a relative metric anyway. You don't even need to scale it to meters. You can just all look at it relatively, how it differs from the rest of the average of the population. Yeah, but for sure you can do that. Yeah, definitely.
Great. So we had talked a little bit before this, and you were talking a little bit about why it was that you found sperm whales such a fascinating study to work with, species, excuse me. I know you mentioned that now the work that you're doing is a little bit different. You're working with some other species. So something you could talk first a little bit about sort of your experience getting to where you are today, both getting to this kind of research and as a PhD fellow now. And this is really because as student members at large, we're trying to get information out there for students and people that are interested in getting to this kind of career area that you are. So I wonder if you can talk a little about that. And then I love the story that you were talking about with the sperm whales and why you found them so especially exciting to work with. So you could talk about that a little bit too.
All right, thank you for the question. So basically I started by looking into internships. I think this is a common way to get into, like, I knew that I'm fascinated by whales and cetaceans overall, and I want to work with them. So I looked for an opportunity during my bachelor's for an Erasmus. Here in Europe, we have Erasmus studies. So it was an Erasmus internship. It's like a grant you can get in all your study cycles, actually. So I looked into that, and I came to Iceland, where I'm based now, and I did my first internship here for 2 months, my first contact with whales. And yeah, I met some people here that ended up 6 years later being my supervisor here, and it was a nice contact. And then I went to, my bachelor's was in biology, just general biology. Then I did this internship, a little bit more experience in the field, then moved on to do marine biology masters in Sweden, in Stockholm. And then I looked into like internships again, and I contacted some people that I was interested in working with. And through the Erasmus again, I went to Denmark, and I met Frederick Christensen, who's also the senior author of this project and my supervisor currently now on my PhD. And I just did an internship with him, and I was really interested in his work. He has developed this whole method. He's like great, and his papers are really thorough and very, it explained very well all the methodology. And so I was really interested in that. And then it happened that, this project came up, and I was able to work on it, learn a little bit more, get some experience, and then continue on now with my supervisor here in Iceland and Frederick together to do a PhD using drones. I'm also using similar methods now. I will maybe expand it to do some other things, but basically step by step. But that was kind of my story. And I really like sperm whales in a way that they combine basically the best of both worlds, in my opinion, like the large size of baleen whales and the social aspect and very interesting social life of odontocytes. So, and they're very big, and they are also living in social groups. So I really, really like, these animals. They have a little special spot for me. But I'm also extremely happy to work with 5 species now on my PhD, from porpoises all the way to blue whales, and looking at size differences also on those ones. It's really fascinating, and all of them are amazing. I don't know. I hope, this was
that's wonderful. Yes, no, that's exactly. It's just helpful for students to hear a little bit about the process and know some of the opportunities that they can look out for. Just that reminder that, unfortunately, that searching for internships and finding those kinds of opportunities is really, really the way, it seems like, for a lot of folks to make those connections.
And so basically, just emailing people, even persisting a little bit, like maybe send a second email, follow up if you then don't get a reply, or also make it personal to them, like show that you are interested in their work, you've read their work. Because some people just have one email. Everyone is kind of doing this, like have one email and you just send it to many people, but it's nice to tailor it a little bit more to the specific researchers. I think this will go a long way.
Makes sense.
A little advice.
It's wonderful. We do have another question that's come up from Catherine McCoy, who's asking, hi, Maria. Did you find any significant differences in volume to density estimates for pregnant individuals? I don't know if that's something you were able to capture at all in your research.
No, we didn't. We didn't look at any. I think we didn't have any pregnant ones, but we never know. There wasn't any. We didn't look into that, no. And I think it's actually quite hard to tell if an individual is pregnant from the drone. I'm not sure. I think there are people studying it now and trying to, develop a method how to actually see if they're pregnant or not. That's very interesting. But I think, for us, it wasn't no, we didn't look into that. And we didn't look at least at one. We didn't have any individuals that might be pregnant. I don't know. We're very unsure. But it's a good question. It's very interesting. It's a very interesting point.
An open research question then.
That's really nice, actually. I'm sure it would change because it has a fetus inside. So maybe it will be as a I'm thinking about that. That's nice. Food. I will think about this. It's very interesting.
Yeah, if that has any applications to the work you're doing now too, I don't know. I think there's just another, right, there's always these other factors that we have to consider, especially if it's hard to tell from just looking at the animals.
Yes, for sure.
Okay, well, I think given that we don't have any other questions, and we want to be cognizant of people's time, I think we're probably going to wrap it up right here. Say thank you again, Maria, for taking time. I know we were talking, you're in a busy field season right now, so we really appreciate you taking the time. Oh wait, I see a raised hand. No, was that a mistake? The person who's raised their hand, if you have a question, by all means, please post them in the Q&A. That's the only way that we can see what you're interested in. Okay, we have a thank you very much from Catherine McCoy, who asked the question about the pregnant and density estimates. So
Thank you.
Perhaps you know her. Yes, so thank you again so much for this really interesting work, for presenting it to us, for sharing your research. Reminder again for folks that the paper is available via open access, that we hold these webinars just about every month. We advertise them on Facebook and on Instagram and through the monthly emails. And that next month, we're actually going to have another talk about drones, but this time in seals and looking at pupping and weather conditions and things like that. So that should be very interesting. We'll have more information out about that soon. We've had a shortage of pinniped papers recently, so it'll be nice to bring that in a little bit. Maria, is there anything else that you wanted to add, or?
No, just I want to thank you for inviting me. It was really nice. I hope, people found it interesting, and I'm looking forward to see the next presentations for the other researchers. Yeah, very exciting.
Absolutely. Wonderful. Well, thank you so much. Thank you all for listening, and hopefully we'll see many of you next month.